Beyond just cells, what other new therapies could hold promise? And so we hope to answer that question.
There's Alzheimer's, and then there's dementia more broadly.
I'm here at the old CP24/Sedigh TV building. How do you separate fact from fiction in a world full of jargon and often a lot of bad actors? That question is all too pressing in the world of stem cell therapies, including when it comes to brain-related diseases like Alzheimer's and Parkinson's disease. The world of stem cell therapies holds a lot of promise for these diseases, but there are lots of caveats. And so we hope to answer that question throughout this video, that initial question that we posed. But who are we? I'm Parmin Sedigh, an undergraduate student at the University of Toronto. And this is
Paul Knoepfler, a professor at UC Davis School of Medicine and a stem cell and cancer researcher.
Let's begin with an overview of the diseases we'll be covering today: Alzheimer's and Parkinson's disease. What are the main parts of the brain that are impacted by these diseases, and how?
Yeah, that's a great question. I think there's some similarities and some differences. So there's Alzheimer's, and then there's dementia more broadly. And so even within just that sort of subgroup, different parts of the brain can be affected. Like, my understanding is with the variety of causes of dementia, it can affect almost any region of the brain. And sometimes that can be due to things like stroke. Like some people end up getting lots of little small strokes, and that can happen in many different places. Whereas with Alzheimer's proper, I think it mainly begins in the hippocampus, and then ultimately it also can affect a variety of different brain regions. So with Parkinson's, it's more focal. It's more like these specific dopaminergic neurons in the brain. So the neurons that are making dopamine in a region called the substantia nigra. And so you're losing neurons just there, but that's having a wide variety of effects because the dopamine can have different impacts on different regions.
What are the main brain cell types impacted by these diseases?
Yeah, so while neurons seem to be the primary cells affected in these conditions, the brain has a variety of other kinds of cells, including glia and oligodendrocytes are two main other kinds of cells. So these have different kinds of functions. They don't seem to be as directly affected by these conditions, but they can be indirectly impacted. Like, there can be regions in the brain with these conditions where you have some inflammation, or even the cell death of neurons can indirectly then potentially cause negative impacts, say on oligodendrocytes or glia. Those cells could potentially also not survive well or not function properly. And even within the category of what we call neurons, it's thought there could be a few hundred different types of neurons. So that also is something to think about. Even though these conditions share, the mechanism of affecting neurons, there are probably many different kinds of neurons that are affected, and some are less affected.
I'm at a statue by Julia Doe, highlighting the ubiquity of mental health illnesses and challenges. That's not what we're focusing on today, but there are scientists that use stem cells to model mental illnesses in the lab and study them closely. Maybe that's something we can talk about next time. Back to our topic and focus for today. Are there neural stem cells that could be mobilized or triggered to repair damage in the brain? Or would all the cells need to be new ones that are transplanted?
That's also a great question. So it's actually been debated for decades whether or not humans have stem cells in their brain, like true stem cells in the adult. And I think the consensus at this point is that there are adult stem cells present in the human brain, but it's a relatively small population, very tiny population. In fact, if you compare it to, say, how many there are during fetal development and things like that, or even, in early childhood or something like that, there's a lot more stem cells in there. And those stem cells sort of earlier in our existence are doing things we might want to achieve with regenerative medicine. Like earlier in development, they're growing the brain, expanding different brain regions. So unfortunately in the adult, like us, even though we probably have small populations of stem cells in our brains, it's not clear that they're actually there for repair. There's not very good evidence for that. Sort of an intriguing question, like, why are they even there if they're not necessarily fixing things? And so it could be that they are fixing things, but in ways we don't really fully understand. The other interesting thing about these populations is that they seem to be only in certain regions of the adult brain. And one of those is actually related to the sense of smell. So one idea I've heard is that these populations of adult stem cells in the human brain might actually somehow relate to maintaining plasticity in our olfactory system as an adult, more so than, say, repairing our brain if we have some kind of head injury or disease. So one of the ideas with regenerative medicine for brain conditions is to actually put neural stem cells into the brain. And the hope would be that they might sort of repair things, make a healthier environment for the other cells that are already there. But those cells also are proliferative cells, so there could be risks associated with that. And so I think the more common approach is to actually take stem cells in the lab and make neurons, for example, in the lab, and then transplant either those neurons or sort of an intermediate population like progenitor cells into the brain. And then those cells have more limited proliferative capacity, and they could kind of make a home in there, maybe grow just a little bit and do what we call engrafting, which again is making like a long-term home in the brain to do something useful.
Based on what we've talked about so far, what's the underlying theory behind how stem cells could help with these diseases?
Yeah, I think the main idea is replacement, the one that I've seen. And so say in the case of like Parkinson's, we've lost our dopaminergic neurons to a large extent. And so if we can replace those and we get new cells in our brain making dopamine in the right place, we could have a net positive effect. So sort of a similar kind of thing is invoked oftentimes, even with like dementia and Alzheimer's, get some new neurons in there, replace the old ones that are either maybe like senescent or dead, and we could improve brain function in that way. But I would say the main other hypothesis is that instead of necessarily replacing cells, we want to make the cells that are already there happier and healthier. And so in this sort of alternate scenario, you would transplant cells into the brain with the hope that those cells, perhaps only being there transiently, would, say, secrete growth factors and have other effects that make the existing sort of original cells in the brain more functional, happier, less inclined to die, let's say. And on the sort of anti-inflammation growth factor secretion kind of side, we do see a lot of talk about different kinds of cells would be used potentially for that, like mesenchymal kind of cells might be sort of anti-inflammatory, things like that. And on the cell replacement side, we tend to see more like pluripotent stem cells being used in the lab to make neuroprogenitors, to then put into the brain.
What are some of the hurdles that need to be overcome before stem cell-derived therapies could one day help treat or perhaps cure brain diseases like Alzheimer's and Parkinson's disease?
Right, so we have this thing called the blood-brain barrier, or BBB, which is there to protect our brain from toxins and other things like that that might be circulating in the blood and get throughout the rest of the body, but we don't want those things in our actual brain tissue. So that's a good thing that we have that, that's important for survival, but it does make for a challenge to get cells or other materials into the brain as a type of regenerative medicine. And then also just the skull, is an obstacle to deliver things. So if we compare trying to do regenerative medicine, say on the brain versus something like the heart, there are other challenges that one might face in delivering something to the heart, but there's no skull around it, right? There is the rib cage and things, but there are ways to get around that. What this means is if we're trying to treat something like dementia or Parkinson's, or even other things we might talk about in future videos like Huntington's or ALS, we have to get more creative with the delivery approach. And so there are two sort of blunt force kind of ways, if one wants to put it that way, to try to deliver things to the brain. And one of them is just to drill a hole in the skull and use a long, fine needle and inject the cells exactly where you want them to be. And that sounds kind of intense, but from what I understand, it's actually not that bad a procedure, and the recovery is pretty good. And in that way, the cells can be put exactly where you want them. Another one is to go through the spine and do what's called intrathecal injection. And so it's kind of reminiscent of like a spinal tap, but you're actually putting stuff in instead of taking cerebral spinal fluid out. And that's a much less precise method, the intrathecal one, because you're just putting cells in there and they kind of will percolate around both in the spinal cord space and in the brain. So there are definite potential downsides to that if you're trying to treat specifically a brain condition. Another approach that's gained interest more recently is called intranasal delivery. And so it turns out the interior of the nose is actually very proximal to the brain. And there's just some membranes in there. And if you formulate the stem cells or other kind of cells in a solution that is amenable to transiting those membranes, basically you have the patient snort the stem cells, or you use something kind of like a nasal inhaler. The cells go to the back of the nose and in some cases may actually go into the brain. And so that's something that sometimes kind of blows my mind, just because of it reminds me how the nose, the interior of the nose is so close, to the actual brain tissue. So you can imagine there are pros and cons to that as well, because again, you may not want the stem cells to be in the part of the brain that is closest to the nose, right? Like if you're having dementia or Parkinson's or other conditions, getting the cells right there in, I guess, the more frontal part of the brain adjacent to where the interior of the nose is, that may not be so useful, but we still are hearing more about that. And there is evidence the cells can get into the brain that way. So those are some of like a physical kind of approach to get past barriers. But another way that we often hear about is just putting the cells IV into the bloodstream and doing something special to help them transit the blood-brain barrier to go from, just the systemic circulation into the brain. So that's also interesting, but you can't just rely upon an IV infusion of stem cells going to the brain without doing sort of extra steps, because again, usually that blood-brain barrier is pretty hard to transit. It is worth noting in some kinds of brain conditions, I think especially like with stroke and other conditions, I'm not so sure about like dementia, the blood-brain barrier is not very intact anymore. And so things can get through from just your regular old blood into the brain tissue in some of those conditions. But I'm not so clear on whether or not for the diseases we're talking about today or the conditions we're talking about today, that would really happen very much or enough. This sort of leakiness of the blood-brain barrier may not be enough to get meaningful numbers of cells.
And following up on this theme of delivery, would a single injection or infusion be enough, or would there need to be multiple throughout a couple of weeks or months or maybe years?
That's another really good question. I think it would depend on the condition. So my sense is that for Alzheimer's and other kinds of dementia, you probably would need repeated injections of cells. So if we think back to the mechanisms, if we're talking about a lot of inflammation and cell death, the cells that you put in are probably not all of them are going to make it. Some of them will. You might get some beneficial effects, but those, my guess would wear off over a period of, say, months or a year or two or something like that. So you'd need another injection. Also, in many cases with dementia, there's widespread damage already. And so you might need to do multiple injections just to get cells into multiple locations where it would be beneficial for you to have new cells in there. With Parkinson's, it's harder to say. It might be also required. Some of the newer research is encouraging that there could be these modest positive effects by a single administration of precursors of dopaminergic neurons, but it's too soon to say like all that research is just really cutting edge. It's happening right now. And so we haven't seen studies of large numbers of patients over many years. So that's what we really need to see to know, can we have an effective treatment or even a cure with one injection? Or do we need, say, an injection every couple years or something like that? My guess is that overall one injection is not too likely to provide a cure in many of these conditions. And so we're probably going to need repeated injections.
Another challenge that's unique to the brain, as opposed to something like the heart, where in theory new cells would be able to eventually function like the old, in the case of the brain, these new cells won't have memories in them. Why is that the case at a biological level? And is there any way to get around that?
That's a really tough challenge, I think, because you could imagine, let's say you have an Alzheimer's patient and they've, just throwing out a number, they've lost 30% of their functional brain tissue, and there is some brain shrinkage associated with Alzheimer's disease. And let's say you had this fantastic new stem cell treatment, say neural progenitor cells or something like that, and you could replace half of that loss. I think that would be amazing. It's not like completely replacing it, but Now you've got this large area of new brain tissue from transplanted cells. Those cells are kind of a clean slate. They've just been made. Say you made the progenitor cells in your lab, you injected them into the brain, they differentiated beautifully into neurons in the right place in the patient's brain. But these are new neurons. They haven't been there, say, in this 75-year-old patient's life. So these neurons actually don't really have memories in them. And so my understanding is that part of the sort of molecular basis of memory is actually combinations of like proteins and things like that. And so these new neurons don't have these molecular bases of memory. And so they wouldn't be the same as the cells they were trying to replace. They would be cells probably without a memory. And so they would then, the hope would be they would integrate, like you had mentioned, and be functional components of this brain, but they would be really different in some ways just because they don't have that, say, collection of proteins that's so crucial for specific memories. On the other hand, they might actually make other cells around them function better, and those existing other cells might have a molecular basis of memory that then could be restored. And so there still could be some restoration of memory, but it wouldn't be intrinsic to the new cells, unfortunately.
I'm here at the old CP24/Sedigh TV building. And this building with a history of news, with that news truck coming out of the side, seems like an apt spot for our next segment, which is going to be on some of the past challenges that the field of stem cell therapies for brain diseases has faced. So first up, one of our questions is that previous therapies have used fetal-derived cells in the past. So is that still something that's being done?
Yeah, there were different approaches that involved fetal tissue. And so there were approaches using fetal cells. And also for Parkinson's, there were fetal tissue transplants, like fetal brain transplants. And then, like you said, there are potential ethical issues there. So my sense is there is still research involving fetal cells, but not as much as maybe in the past. And I don't see as much about fetal tissue transplants like for Parkinson's anymore. So I don't see like a lot of controversy there. There are some promising trials, and this includes for Parkinson's, that involve using human embryonic stem cells. So those are not really fetal cells. It's a much earlier, kind of process. You just get like a 5-day-old human embryo left over from in vitro fertilization, and you can derive these embryonic stem cells from that. So there are no specific tissues in those embryos. They have maybe like 100 cells, something like that. So those cells are not as controversial anymore. I would say like 20 years ago, especially here where I am in the US, people were maybe more evenly split about it. And the most recent polling I saw actually showed that there was like a 2-to-1 favorable opinion amongst the public about the ethical nature of embryonic stem cell research in the US. So I thought that was really encouraging.
Well, another question is the continuing issues with stem cell clinics that continue to tout unproven cell therapies. So how does that work? And could you talk a little bit about that?
Yeah, we've seen, especially in the last 20 years and even more so in the last 5 to 10 years, there's a lot of these unproven clinics that are saying they can use cells, especially mesenchymal cells, to treat a variety of brain conditions, including Alzheimer's and other kinds of dementia, Parkinson's, many other brain conditions. And these clinics, they don't really do clinical trials like we understand them. They don't tend to work with regulators like Health Canada or the FDA and the equivalent European regulators. They just kind of go off on their own and say, hey, it seems like a good idea, say, to use MSCs from bone marrow or fat or maybe other kinds of cells, or one that's growing is perinatal cells, so like umbilical cord MSCs. It seems like kind of a neat idea to use these cells to treat brain conditions, and they just kind of do it, and they don't, they're sort of in uncharted territory because, again, they don't really do clinical trials. So they might see something like, snorting stem cells for dementia also as something that could be very profitable for them as a business because it's fairly straightforward. It's much simpler than, say, trying to inject some cells into the brain or the spinal cord or something like that. And so, although some clinics actually do intrathecal injections as well, unfortunately, which has a higher risk, you can imagine putting cells like right into the spinal cord.
Beyond just cells, what other new therapies could hold promise? For instance, what about gene therapies, maybe in combination with cell therapies?
I'm really interested in gene editing for neurological conditions, and especially things like pediatric brain tumors and cerebral palsy. But it's sort of the beginning of a new area of research, I would say, trying to do these things like gene editing in the brain looks very promising in some ways in like animal studies, but we're just sort of now starting to see more clinical trials in that area. So there's a lot that we can kind of hope for in the future, but also be cautious, I would say, because if you do gene editing, say for a neurological condition, it's essentially like a gene therapy, and you can have different kinds of potential side effects depending on the specific gene that you're targeting, the approach. So like with AAV viruses, we've seen recently, unfortunately, in some very different kinds of work, some patient deaths who received, new, even FDA-approved gene therapy here in the US comes with risks, especially for whatever reason. With these viral delivery approaches, sometimes it can lead to problems in the liver. And so that could be true in the brain too, if we're trying to treat something there. The other thing that's really exciting is combination cell and gene therapies. So in that kind of approach, you're still thinking about, say, delivering cells in the brain of a patient with dementia. But prior to delivery, you've actually CRISPRed those cells. You've gene edited them to be different in some way, like maybe they survive better, or they can replace cells that had some kind of mutation in them for a specific brain condition. And so these new cells now have a healthy copy of that gene. So I think that's a particularly promising area where you can gene edit cells first in the lab and then deliver those to a patient and maybe have better outcomes.
Let's now talk about clinical trials. And let's start with the clinical trials for Parkinson's disease that recently wrapped up using stem cells. Could you talk a bit about those?
There were 2 trials using iPS cells and 1 using human embryonic stem cells. And in the 2 iPS cell trials, one of them is using autologous cells, so the patient's own cells, which is pretty cool. So you don't have to worry about like rejection of the cells that you're going to transplant in the brain. And then the other one is using allogeneic iPS cells, which has its own advantage of maybe like being able to scale up from 1 iPS cell line to make like 1,000 therapies for different people. And then the embryonic stem cell study was also pretty encouraging. So in each case, the main goal with these was really to try to understand the safety and just kind of the properties of the dopaminergic neural precursor cells that are made from the stem cells that are then transplanted. And so all of them had like a good safety profile. You have to worry about like the cells could potentially form tumors, or they could cause new movement disorders, which would be really, frustrating when you're trying to actually fix disorder, symptoms of Parkinson's. You could end up getting more problems. And so with all the trials, they actually look pretty encouraging. Like there weren't severe adverse events related to the transplanted cells, so that was great. And then in each case, there were kind of hints of potential improvements in the symptoms of the participants, the Parkinson's disease patients who were participating. So all of them were small trials. It's kind of early days, but it's really the first, I would say, set of 3 studies that give real hope that stem cell-based therapies could be an effective treatment or even a cure for Parkinson's.
And what about clinical trials for Alzheimer's disease? What is that looking like right now?
I feel like it doesn't seem quite as promising, unfortunately. And again, this might in part reflect the fact that Alzheimer's and dementia are these really complex conditions affecting many regions of the brain. And oftentimes when patients are diagnosed, there's already a lot of damage. And so it's not necessarily just about replacing cells. And like we talked earlier, even if you can successfully replace cells, those cells may not have the same functions. They won't have the same memories as the cells that were lost. So I think it's a tougher situation. And even just like the mechanisms of Alzheimer's and many kinds of dementia are not as well understood as the Parkinson's mechanism. So that makes it harder therapeutically. There was one trial that showed seemingly encouraging results for MSC therapy for Alzheimer's disease, I believe it was. I think it was Alzheimer's and not some other kind of dementia. But there again, I wasn't so clear on the mechanism of how MSCs, and I believe it was IV infusion, how could that really effectively help Alzheimer's? So I'm going to closely watch that one in the future. I think there's going to be follow-up trials. So I think there are some indications of potential hope, but it's just such a, the whole span of different dementia causes. There's the whole Alzheimer's area. It's so complicated that right now I think it's going to be tougher to get an effective regenerative medicine therapy, but maybe in the longer scope of things, we might still see that.
Finally, what is the most important thing that people should take away from this video?
I think the most important thing viewers should take away is that there is real hope from stem cell-based therapies for dementia, including Alzheimer's disease, and especially for Parkinson's disease as well. So we're getting closer as a field to actually making an impact with a new cell therapy for these kinds of conditions. But at the same time, viewers have to think about something that might sound too good to be true. They have to, still be patient in some ways. And I know that can be very hard facing a serious illness. So if we want this longer term hope, especially for like regenerative medicine for dementia, that's going to still be a marathon. And the current generation is probably not going to fully be the ones to figure this out. And so we need younger scientists involved as well.